Okay, I think we're ready to go. Thank you all for joining us here for the Donald Michaels Distinguished Lecture Series. This is the third year of the Michaels Lecture funded by his family in honor of his scientific career. We're happy to have them with us today. If you don't know Don and Vicky already, I'm sure you'll see them around as they've been here, as you all know, many of you know, for many years. We're just going to say many years. So the goal of the series is to showcase research of distinguished scientists like our speaker today, who integrate across levels of biological organization and disciplines to address sort of major questions in marine and coastal science. In the spirit of Dr. Michaels' innovative work throughout his career in the field of sort of crustacean, molecular and cellular physiology. Throughout his career, he continued to visit BML regularly from his position at Colorado State. He brought students with him. He's been a long-standing member of our community, and we're really sort of pleased that he's been able to help sort of continue his legacy here by hosting this seminar speaker. As you know, Dr. Kelly is our third speaker in this series, and Karolina Zabinski will introduce her in just a second. One quick sort of bit of announcement. The whole sort of series involves Dr. Kelly being here for a couple of days. So we'll have this morning's research seminar. Tomorrow there's a community lecture, which will provide a more broadly accessible sort of view of Dr. Kelly's research. And there'll be members of the community, not just the lab, but also outside the outside Bodega community here. And throughout the sort of period of her visit, there'll be opportunities to meet with her, either one-on-one or as members of different groups, to interact through sort of various meetings and social events. So at this point, I'm just going to sort of turn it over to Karolina Zabinski, who's going to introduce Dr. Kelly. But first, let's thank the Michaels for their support of this series. All right, Karolina.
Hi, it's good to see everyone and meet new faces, new people. Hi, Molly. Okay. So Dr. Kelly will describe her research in lots of detail today, but she is an environmental microbiologist and associate researcher at Scripps Institute of Oceanography, and her research aims to understand the critical role microbes play in nutrient recycling and organic matter transformation across coral reef ecosystems, seaweed aquaculture, and intertidal habitats. Okay. In particular, Dr. Kelly leverages marine biogeochemistry with microbial genomics and untargeted metabolomics approaches to understand how foundation species-derived organic substrates shape microbial communities and their ecological functions. Now, I was asked to give a brief summary as to why I nominated Dr. Kelly to join us today. And so personally, I'm excited to have connected with you in person, but our lives have, my life has crossed with Dr. Kelly's lab since 2022. So back in 2022, I went to WSN and attended one of her former graduate students' talks, Zach Quinlan, who is looking at how different foundation species exude these organic compounds and they drive different rates of microbial metabolism. And I was like, wow, that's so cool. Like, do you want to grab a beer with me and talk? And we ended up creating this side project on seagrass exudates, and this kind of flourished into this collaboration between UC San Diego, UC Davis, and now UC Merced, characterizing the seagrass rhizosphere metabolome in adult plants as well as seeds. And so Dr. Kelly's lab and all of her students have kind of helped teach us at UC Davis about this new field, metabolomics. Additionally, Marcella, one of our colleagues, now is at UC San Diego at Scripps in Dr. Kelly's lab. So there's all these personal connections between our campuses. So naturally, as a biologist, I wanted to increase these collaborations at higher levels of biological organization. So when the Michaels Lab seminar came out, when they were seeking nominations, I thought Linda Kelly would be a great person to nominate. So when I think, as a member of Bodega Marine Lab for 5 years now, I see our strengths in our perspectives on ecological and evolutionary feedbacks, our knowledge on the natural history of our systems, and our desire to incorporate interdisciplinary approaches in our work. And as a barnacle or like fouling member on the UCSD SIO community, I see how skilled these folks are at scaling between levels of biological organization and their perspectives of ecosystem processes. Yet, like us, they also aim to incorporate different approaches to answer their questions. So I hope that y'all enjoy this seminar and that this continues building a bridge between our foggy, windy coast and that sunny La Jolla shores. So yeah, thank you. I'll let you take it away from here.
Thank you, Carolina. Well, thank you all for coming. Can you hear me? The mic is on. I feel like I'm in class a little bit, like you're all so far away. This is like very typical, but it's okay. I'm honored to be here. I've never been here before. It's so beautiful and it's just been so peaceful. It's just like since I've gotten here. So thank you for giving me the opportunity to come here, tell you about my research, what I've been doing. This is what I'm going to show you today is the work that's closest to my heart. And so we'll talk a bit about that. I do like to entertain questions, but given like the Zoom link, I think, but like, please stop me if something is, just very unclear. We right back into it. But in my lab, I mostly study, the main thing, we study a lot of things, but I am profoundly interested in how marine macrophytes interact with their seawater environment and change the chemistry of that environment around them. And that could be, I study a lot, you're going to see a lot of coral reef work today, so corals, but also kelp forests, seagrass meadows, intertidal regions, tide pools. They all have this collection of benthic organisms that exude molecules into the surrounding seawater environment and have a profound effect of those habitats and how those habitats, processes within those habitats and how they function. Okay. And so I do use, I got my PhD, I'm an environmental microbiologist by training. My PhD is in a cell and molecular program. So I use a lot of genomics. Metabolomics is a new field. I study dissolved organic matter, so DOM, DOC, and that's so a lot of biogeochemistry. So I've kind of become a biogeochemist, not classically trained, but kind of more on the fly. And that is what we do in my lab. So just to reiterate what I was talking about before, these habitats profoundly affect the surrounding seawater. These are so all of our coastal habitats. These coastal habitats, they're never bare, right? You always have some kind of plants growing there, animals, and they exude a significant amount of their primary production into the surrounding seawater, something in the order of 20 to 30%. As they're photosynthesizing, they just release it out. And we don't understand exudation as well as we would like, but there's ideas about just like overflow metabolism. It's just easier and less expensive just to keep it on and just release what you don't need and just keep cranking. So for example, corals will release a suite of metabolites into the surrounding water. Depends on what coral you are. Let's see if I can get this. So healthy colonies, branching and boulder colonies release different molecules, but also bleached corals release like a distinct suite of molecules, and we can measure them. And so the open ocean has its own kind of suite of molecules, its own metabolome. And then we're also starting to look at how this kind of chemical signaling that happens with coral spawning. I'm not going to talk about it today. I'll talk a little bit about it more at the public lecture, but So with synchrony, like the way they synchronize, like when to release their gametes, we think it's chemically, like kind of like a quorum sensing event, it's chemically sensed. And then also there's a lot of cues that allow the larvae to decide where they're going to settle down and live. And that's all, there's a lot of chemical ecology there that we're interested in as an overview. And I already mentioned this, but just to kind of show you, is that if you put a coral or a macroalgae or seaweed in a bucket, you can measure the release of carbon over time. It does correlate with oxygen, and so they are indeed releasing molecules into the surrounding seawater. And it's a diversity of different types of molecules, and there's so much that we don't know and don't understand about all of this. And it is quite a bit of carbon that comes out of them. And all of this carbon more or less goes into the microbial food web. And so it's roughly about 20% of their daily budget of photosynthesis going straight into the water. And we can see that across different systems, this is a coral reef system. Coral reefs are nice because you often have unilateral water flow going across them. And so some of the physical dynamics are worked out pretty well, where you could track the water through like a Lagrangian approach, and you can see what happens as offshore water moves across the reef. So what we often see is that as you move from offshore across the floor reef into the back reef in the lagoon, is that you get a depletion of dissolved organic carbon. And this has always really confounded us, because you're moving water across a coral reef, which is just, it's all primary producers. And we know that they're releasing DOC into the water all the time. So why are we getting a net drawdown of that DOC? It's always confounded us. And then we also tend to see an increase of nutrients as you go across the reef as well. And I'm showing you nitrate here, but you do tend to see a net increase in phosphate. Because I come from a background so this Moorea is a high island, and so when I run into my colleagues, they always think of the land. Everything is about the land. But when I was a PhD student, I studied atolls in the middle of the ocean, and there is no land, and you still get an enrichment of organic nutrients. And so I always thought, I was like, no, it's the reef, it's not the land. And so we've always kind of intersected in that way, where they come more of like from a chemical lens, and I come from a biological lens of how these processes are happening. And so here, just on the very right, I have these just to show you, is that these are the types of molecules that we're starting to identify and classify using the mass spectrometry, so the metabolomics. So we're also looking at all these different classes of molecules, classes of organic molecules that change across these seascapes. And that is what we've been really excited about. And so how do we do this? It is complicated to try to understand chemistry in a complete ecosystem, like a complex, diverse system like a coral reef. There's a lot going on. You've got the island there, you've got the reef there, you've got the fish there. And so to start out, we really wanted to ask, what do different primary producers make? Do they make like kind of very different types of molecules or classes of molecules? And it's also a way that we can build a database. So if we can identify that like this coral genera makes this suite of molecules, and I'm going to show you a lot of slides about this, so this is kind of like a little preview, and we could go back to it, but if we can start to build like a very nice library of what the different functional groups make or primary producers make, then we can maybe get a better handle when we find them on the reef. We can say, well, we think that Dictyota is making this molecule. So that is what we've set out to do. We have We do a lot of pumping. We have these peristaltic pumps that have like 32 channels, and so when I go to the field, I pump like thousands of liters of water through all of these tiny little expensive filters and cartridges. And so when my family asks me, they're like, "What do you do out there?" I was like, "Oh, filter water through really expensive filters, and then we put the filters in the freezer." So like schlepping water is like what I end up doing when I'm in these exotic places, just like schlepping, 20-liter carboys, like 30 of them off the boat and into the lab. It's very glamorous. But we do these exudations in these very clean polycarbonate containers. And then we do solid phase extraction to concentrate the DOM, and then it also helps us desalt it so that it can go into the mass spectrometer. And we cannot capture 100% of all the molecules, but we are getting about a third of them onto the PPL. So this is what it looks like when it goes through the mass spectrometer, and some of the types of questions that we can ask at the back end of the mass spec run is that, so what are the different types of compounds that are there? Like, are there a lot of amino acids? Or we're looking at like a lot of lipids and steroids. How abundant are each of these molecules? Like, so in the DOM pool, are there some classes that are making up like a big proportion of what's there? How do they vary across time and space? So if you're doing a spatial survey, those are questions that you can ask. And essentially, the way to think about this is that we've taken these chemical assays and approaches, if you will, and then we've looked at them like an ecologist would look at them. You're going to kind of see that, and you might imagine that it's not super easy to interact with the natural products community at Scripps. They don't understand why I want to look at 1,000 molecules and not just one. But we are getting along, and I listen to them, and they listen to me grudgingly. And then, of course, like I mentioned, what do the different organisms make? And so you can see so this is just kind of like the run through. So first, the sample, it goes through the LC. And then, so you get MS1s, which really just give you mass. So that's mass and retention time. But really, what is the advantage of what we do is it's called tandem mass spectrometry, and each peak in the MS1 gets fragmented, and those fragmentation patterns are a way that we can move to looking at structure based on their fragmentation. So it's almost like handedness. So you can see, and you can match them up. Because when you get a database match just based on mass, it's not very easy to tell the shape. You can often get, if you get a really good match, you can get an elemental formula, but it's very hard to get that structural formula that actually allows you to call it an amino acid versus a steroid. So, and you see in this color plot over here, that so everything in blue is like very low, and so this is our water control. You can see it's very empty. It has a few things in it. But then you could see for this macroalgae right here is that it makes these suite of molecules, and essentially nobody else makes them. Same over here for the turf algae. And then we also are using the peak area to estimate relative abundance, and so you can look at the magnitude of those as well. But by and large, organisms make a lot of unique features, and that's what I'm going to on the next slide. So here, if you were to kind of take this list here, right? These are all enriched in the Passalopera. It's a type of coral. And so what it does is that, in an ordination, you can really see how they all separate, the different primary producers, right? And so it is this kind of list that's driving that. But you can see that what Passalopera has made, basically no one else made it, not even the other coral. And so it is and this is what I was talking about, is that what we've seen when we did this study, and kind of this Venn diagram is also showing you, these are all the unique molecules made by each organism. And then these are like the cosmopolitan ones. There's really not a lot in there. And so in that way, then I felt I did have more confidence that I could look out on the reef, and I could find these molecules, and I could say, those are Dictyota are making these molecules. And so that is where we're kind of moving towards, and that feels like a very powerful way to really understand like how the benthic community directly interacts with the microbial community. Microbes are metabolizing those metabolites, and really just understanding those connections and those relationships. So another really cool thing that we found by being able to characterize the DOM is that we can actually see that the different organisms enrich the seawater environment with different nutrients. And they are mostly releasing like organic forms of, so dissolved organic nitrogen and dissolved organic phosphorus, which very rapidly gets transformed by the microbial community, so that you end up getting, I think that this is what contributes to that enrichment of nitrogen or nitrate and phosphate on the reef, is all this remineralization of the organic nutrients that are released by these primary producers. So this is like since the 50s, we've talked about, how coral reefs are they're very good at nutrient recycling. It's like, how does that work? And from my perspective, from my lens, it works because the corals are, they're drawing in the ocean water. They're both plant and animal, so they're eating all of the plankton that comes in, and then they're just like explode, and then they're releasing just a bunch of dissolved organic nutrients into the surrounding environment that all the other organisms can use. They can't, the algae can't directly feed on basically the chemostat of ocean that keeps that hits the reef over and over. So we had increased nitrogen in Passalopera and in turf algae. The turf algae, that's probably from cyanobacteria. But all of the algae are very low in phosphorus, for example. So only the corals really can contribute a phosphorus signal. And so what's really nice, what was nice to see is that this chart is from the annotations of the mass spectrometry features, right? So this is like we annotated the elemental formulas, and we derived this like phosphorus to carbon and nitrogen to carbon. And these two plots are our bulk analyses from the water that we ran on the analytical lab. So we did get a good correlation from the amount of total nitrogen and total phosphorus with the amount we were measuring being released by the organisms using the mass spectrometry. So that was really cool to see. Okay, and so this is just a little bit of more, it's like, I guess it's kind of like as a teaching aspect, is that this is the way that we are able to identify, to give a name to a lot of these compounds like tryptophan or carnitine, or the different steroids that we find, or the different benzenoids or polyphenols. Is that because, so it's two things. The fragmentation patterns allow us to do these matching here. This is called a mirror plot. And so this is the fragmentation pattern of these two molecules So even if your mass, like your machine might not be precise enough with a mass to charge ratio to get like a perfect mass that you feel confidence in. And you could also, you get a lot of different wiggling around with different like functional groups. There's a lot of diversity in the molecules. They just might have an extra hydrogen, and so suddenly the mass doesn't match anymore. But when you fragment it, all the pieces of it match. And you're like, that is a structurally related molecule. And so what we do is we build these networks that are based on the fragmentation patterns. And when they're very close together like this, they are structurally related molecules, but they just might have like a little additive that's a little different. But by and large, you could think of them as being functionally similar or molecular families. And so that's been really powerful. And from an ecological perspective, is that if you're only able to ID like a handful of molecules, you're kind of stuck. And that using the molecular networking, we're really able to get maybe like 30, 42% of good matches that we feel confident about. So that has been, and it's changing, it's always getting better. The cheminformatics are improving just as you would expect, and so that is going really well. And so we're really happy with that. And you can't identify everything that was in there. I still feel like I have a lot of blind spots, but we're really making a lot of advancements. It's going really well. So this is what the network looks like from the study that I've been showing you with those 5 primary producers. And they're color-coded by organism. And so this big orange network is made by Crestus coralline algae. Crestus coralline algae makes the strangest molecules. We cannot identify them. It's really frustrating because we use Crestus coralline algae in a restoration setting for coral reefs. And so we would like to know what CCA exudes that smells so wonderful to larvae. And we can't annotate almost anything from the CCA. They're all very far known. And this is one of them, but only CCA makes it. So all of these nodes is like a diverse assemblage. It makes, we get thousands of features per organism, but 1% of what it made was this one, this network, subnetwork. We don't know what it is. But we do have ones that we can ID. So this coral, it's a Poeciloptera. It also made it a pretty high abundance. And it's a halogenated tyrosine. So it's an amino acid. It makes quite a bit of it. And it's a very fun thing that we're like, this coral makes it. I'm going to show you how we determine whether it's bioavailable to microbes and kind of the next set. It's an amino acid. It's delicious. So Dictyota, these brown algae, make a ton of these, like kind of prenol lipids, like terpenes and retinoids, and they actually make a ton of them. This is 5% of all that we measured in the machine. It's only made by Dictyota. So it's just been really fun just kind of seeing all the different things that the primary producers make and what we think that means ecologically in the system. All right, so this is Zach, who Carolina mentioned. Zach was my first PhD student. He was at the University of Hawaii as an undergrad, and I worked with him there a little bit, and then he came to my lab. He was outstanding. He is outstanding. I miss him. And but he set up an experiment to look at the bioavailability or the lability of all of these molecules. So what do they make that can be eaten by microbes, and then also what are all the features that we would call recalcitrant to microbes? And which is becoming, the recalcitrant part is kind of a big thing happening now, especially with seaweed aquaculture, is from a carbon sequestration lens, is that if you can understand all of the recalcitrant features, you can estimate you know, how much potential carbon sequestration you could be having. And so they're both interesting. The bioavailable ones are interesting if you want to understand, you know, connections with the microbial community and these habitats. But recently, there's a lot of interest and enthusiasm in the recalcitrant ones. And the way we run these experiments is that we do the release, just like I explained before. Then we, after we do the release, we always refilter the exudates. So you can think of them as, there's no microbes in that portion. You think of it as just food or dissolved food. So like this one, this is Porites food, 2/3 volume. And then we put 1/3 of just a natural, like the seawater or reef water inoculum. And then we run their batch cultures. So then they, so since they're a little diluted, they grow for a little while in log phase. They move to stationary, and then we harvest them. And Carolina brought up a little bit about like kind of the growth curves. And so we've done a lot of work. I've done a lot of work looking at like how primary producers elicit different types of microbial growth. Some of them release a lot of DOM that's really labile, and the microbes grow really fast, and so that tends to be like fleshy algae. Whereas some corals kind of, they don't release very much, and also CCA, and the microbes just kind of like diminish. So just kind of thinking about, you know, habitats and kind of their respiration. We have looked a lot at that and how different functional groups kind of elicit these different types of metabolisms in the water. But here, I'm really going to just focus on the metabolites and just show you how we estimate which exometabolites are bioavailable to microbes and which ones aren't. And we do these in replicated experiments. So this is just statistically looked at. We just look at the drawdown from the T0 to the end. And if they had a significant drawdown within the treatments, the replicated treatments, we classified these as bioavailable or as labile. And then if they did not pass, like our different thresholds, we would consider that molecule recalcitrant. And this is just another example of, we have a lot of what we see is that you really only get the signal if the primary producer made it, right? There's also, so if they didn't make it, you always have like a little background, because all of this is done in the natural reef water that's in there. There's all of these molecules are in that background water, actually. So that's like a lot of what we do are these pipelines to kind of, we also have to kind of ignore all of that background. And you actually see that a lot of the things that we identify as these like exudates or specific exometabolites, if you just scoop up a bucket of water, you will find all of those in like a little bit in there. They are all still residing in there. This is an example of some of the different types of molecules that we see and whether they're labile or recalcitrant. And we can look at their different names. And so one of the things that that really resonated with us is that it wasn't really like, the labile ones weren't just a lot of carbohydrates or, or amino acids or organic acids. We had labile compounds all over the chemie tree. All different types of classes were bioavailable to microbes, just in the lipid category, in the steroid category, benzene groups that were just really surprising, but also like a lot of the compounds that contained nitrogen and phosphorus were also bioavailable. But then also we can start looking at what types of molecules are made by primary producers that are recalcitrant. And these are the ones that would be of interest from a sequestration. Looking at which types of primary producers make more recalcitrant compounds, those are the ones that are not going to just get immediately respired back into the environment. So we're going to transition a little from the chemistry to the microbes. Microbes are like what the microbes, like their function on the reef is like what keeps me up at night. That is what I want to know. I can see these signals of different microbes, and I want to know how they're interacting with the habitat. I want to understand what they're doing. I want to know what they're eating, and I want to know how they're kind of helping their community. And those are all the things that I look at. And so I am profoundly interested in how all of these major processes that we think about in all coastal habitats, but particularly in coral reefs, these processes like production, respiration, calcification, nutrient retention and recycling, and particularly important in coral reefs is just net accretion. A reef needs to be building. It has to be net accreting, not net dissolving to stay a reef. And then sustaining this incredibly high biodiversity of consumers that we see on reefs. Like, how does all this happen on reefs? So humans have been fascinated about coral reefs since they've been able to see them and see how much is going on there. And all of these, like, what is like, from my perspective, it's all invisible. What is turning all of these like gears on the reef. All of these interactions, it's all the chemical ecology, the exchange of metabolites between organisms, and the workhorse are the microbes. They are the ones that are interacting with what the primary producers release into the water and what comes out the back end of that. So when I finished grad school, I had the opportunity to engage with this program, this monitoring program that NOAA does with all of these are our US Protectorate islands. So they include the main Hawaiian Islands and the Northwest Hawaiian Islands, which are uninhabited. The Northern Marianas, there's quite a few islands there. We have these, they're called PRIA. They're these remote island atolls, Pacific remote island atolls. And that is these ones in the middle. These are some of them are the Line Islands. Where are my lines? Oh, they're Kingman, Palmyra, and Jervis. So then we have these Johnston and Wake, and you can probably, I'm sure a lot of you know, these are strategic territories. They did a lot of like testing out there, and there's a lot of military presence, especially Johnston. There's a lot of bases out there. And then American Samoa. 35 islands, and then so on a 3-year cycle, NOAA goes to each island. So they do, a third of them each year. And so I have a student out for 3 months out of the year. So at least one graduate student would do like one leg, so 1 month. And so we monitored the microbial and chemistry side of this. We partnered with NOAA for about 10 years, and I ran the program, I led the program for 3. And I'm going to show you a microbial data set that I got. And so what it's given me is that I wanted to understand what are the microbes that are enriched that are important on coral reefs. It is not that easy to understand which microbes come from where, right? Because so it's just water, right? So if you go and you collect water from a reef, there's a huge ocean there. That ocean is just like hammering the reef all day long. And so how do you know which ones is just from the ocean and which ones are actually reef enriched or coming from the reef or important on the reef? So this gave me the opportunity. We had a big data set where we had collected just a sample just in the water column at 5 meters. and then a sample really close to the reef at 10 meters, so right on the reef. And we think we kind of like almost try to kind of suck it on the reef like within the boundary layer, just to really get the reefy bugs. So, and I call these bugs that are my reef bugs the usual suspects. These are classes of bugs or families of bugs that tend to be enriched on the reef compared to the ocean water. And so we did this comparison that was between that 5-meter sample and our reef sample. And so if you look at the middle, this is this kind of the log2 fold change of the benthos to the water. But so here, if you see the ones at the bottom that are in green, those of you who are microbial oceanographers, I don't know how many of them I have in here, but these, you will see these are our water bugs, These are our cyanobacteria, that's our Prochlorococcus and Synechococcus, all of our SARS. And so these are very watery bugs. And you can see that we can see like an enrichment here in the water one. And then up here, where we got good kind of enrichment on the reef, these are our usual suspects. So we do tend to see a lot of Rhodobacter enriched on the reef, and more Flavobacteria, clades of microbes that tend to that are good degraders of DOM. But there's just been these really interesting ones that I was like, what are you doing? We've got these Rhizobiobia. They're related to the nitrogen fixers in the plants. You see a lot of those on the reef. And some of these like methylotrophs, these are single carbon organisms. So just kind of just a lot of interesting metabolism that all I want to do is figure out what they're doing there and what's their role. And so I started a project about 5 years ago where we wanted to look, we had a sense that we could also see that different processes happen at different times on coral reefs. So they're all autotrophs, so a lot happens during the day. And that we got a snapshot of seeing that we could measure discernible differences between the day communities and the night communities. And because it's so tightly linked to processes of photosynthesis and exudation on the reef, we thought that that could really help us understand what the organisms were doing, the different microbes or the different usual suspects were doing. And so, the first time we did it, we only had like a midnight sample and like a daytime, like a 10 a.m. sample. And so we couldn't really understand much. We were like, well, the ones at night, like, are they growing in the night or are they leftovers from the day? Like, I didn't know. I was like, is the night one actually a day signal and the morning one actually a night signal? And so we built these autonomous samplers because it is not safe to go out on the coral reef in the middle of the night. It's sharky out there. So they do collect, we set them up so they could collect time points. So we collected 8 time points from the reef and through a Sterivex filter into a clean bag, and we can measure these different analytes. So we measure different nutrient concentrations, cell composition and abundances, and then we could also do the metabolomics on that water. And so I'm just going to show you a little bit of some of the patterns that we saw with some of the usual suspects. We could see that we did have some patterns with the different taxa. So with the Flavobacteria, they tended to be more prominent in the morning and they dipped down in the afternoon. Where these Rhodobacteria, we always see that the Rhodobacteria or the Rhodobacters tend to come up in the afternoon and become very prevalent into the early evening. And so, we're starting to try to understand, like, if we can pick out a few taxa that look very promising to have this kind of diel signal, and whether we could also pair that with the metabolomes to see if we can better understand how they're functioning on the reef. So we have some leads. This is all kind of early days. We have no nail in the coffins, but we do have examples of bugs. Like, this is an ocean spirelli. It's a nitrifying bacteria. It looks like it goes up. It's not growing very well in the night, and it looks like it grows during the day when all of the metabolism is happening on the reef. And we can also see that with our water chemistry that we do also see patterns. We see that we get, in the afternoon, we have higher DON, higher ammonia, lower nitrate, which is potentially signaling just remineralization. And we also have certain metabolites that we can see, that we have like nice little spikes. So this is a good 3 p.m. spike. And about 2 or 3 p.m. is kind of when your peak P/R is happening on the coral reef. So you have a lot of production happening during that time. And so this is kind of where I'm at right now. I have moved to a new project I did just last year. I got my NSF career awarded, which was incredible. Thank you. And not only did I get the career awarded, which is incredible, but I got it's to do like what is my favorite project, to continue understanding what the usual suspects are. And so so I'm at a place where I was like, I need to sequence their genomes. I have a lot of surveys from different times of the reef where I can see that, like, this specific ecotype or OTU is like 20%, and I could go in and sequence that, do a deep sequence, and then I can assemble those genomes with as a metagenome assembled genome. And so just looking at their capacity, their genomic capacity, and then comparing that to the way we see it being present spatially and temporally on the reef, and then you can't culture any of these bugs, right? But just I can do some transcriptomics potentially in an experimental setting and really seeing if I can iron out some of these metabolisms. So the first one that we put together was this. And so I have it represented as the family, but the mag that we created is where I have one, it's an OTU, it's not an ASB, it's 97% similar, but it's represented about 20 to 30% in the sample. It's a very dominant bug or a population, right? And so we assembled that one. It always came back as an unclassified Rhodobacter ACA. We could never identify it by the 16S. But at the whole genome, it's pretty closely related to this Phaeobacter italicus, which is well known for as a DMSP breakdown site. So it's not, that makes a lot of sense. Corals release, the symbionts inside corals release a lot of DMSP, but we've never, this bug is really abundant and it's really dynamic on the reef. We've never really, and it has all these implications for sulfur cycling on the reef, and the way that it breaks down sulfur, there's 2 different ways you can break down DMSP. One releases a lot of dimethyl sulfide and the other one doesn't. So I think understanding how this is working has not been done at all. They're also very interesting because they make antibiotics, so they produce TDA, which is an antimicrobial compound, which is really interesting. That a reef would harness a bug that could potentially be playing chemical warfare on the reef. And as of now, we haven't found the TDA in this genome. And they also release auxin, so plant hormone production. So very well known. They have a few different secondary metabolites that induce plant growth. So just thinking about that dynamic on the reef, or even for corals, how they can be playing a role, is really exciting. And so this is just kind of the first one that we've put together. So I want to thank you again for being here and listening to me. It's been great to come up here. It's been great for me. I feel so honored. Thank you, Don and Vicki, for just having this great idea to bring lectures up. I'm so thankful, and I'm thankful to Jay and Carolina. Thank you for nominating me and bringing me up. It's so wonderful. And then I worked with Ellie and Jennifer. They helped getting me here with all the logistics, and were very patient with me. And I'm grateful for that. So thank you so much. And I work with, I have really great collaborators, a very wonderful support group that I work with the chemistry team, and my coral reef systems group that I've been working with since grad school. I have wonderful students. I can't thank them enough. Like, they're the reason I get up in the morning. And yeah, it's a good life. Thank you.